Hi, this is Nick Cap, and this is Biology 240. This is microbiology, and we're going to be looking at Chapter 9, Microbial Growth. Again, remember that these lectures are meant to supplement and highlight what we read in the book, and during class sessions, we will also discuss a number of these things. So make sure that you go to the class sessions in order to look at that. What we see is that what this picture is showing here is that we microbiologists are changing the way that we look at microbial growth. In the past, we really kind of looked at a monoculture. And so you would try as hard as you could to get your bacteria in just one form, with one genetic form, what we call a monoculture, just one type of bacteria, one genus, species, one particular strain. This worked really well when we were trying to work to cure diseases, because there's usually one bacteria that would be responsible for curing a disease. And we were able to identify that, we were able to look at that, we were able to get rid of it, and then we cured a disease. But in reality, microbes don't necessarily grow in a monoculture. They grow in a mixed culture of what we call a biofilm. And so on this slide here, it just shows you a little bit of kind of what's going on in biofilm. You have a bunch of cells and you have a whole bunch of extracellular matrix. And this is kind of the scum on the side of this kind of rain barrel right here. And that's something what you'd see. And that's normally what we would see for growth. And so in Chapter 9, we're going to be looking at growth. A lot of it is monocultural, but we may be in the future kind of looking at this mixture. What you should know after reviewing this materials is we're going to be looking at the generation time of microorganisms, be able to explain and plot a growth curve. You want to compare and contrast different ways to count microbes. You want to compare and contrast to discuss ways of how cells multiply. Again, you need to talk about what a biofilm is, what are the economic and health ramifications of biofilms, and explain what this thing that we call quorum sensing. And quorum sensing is a lot like this. You'll have a group of people together and it's like, okay, let's all do one thing at once. And bacteria can actually do that as well. And the big thing when we look at bacteria is this thing that we call binary fission. And what we see on the left-hand slide over here under A, we have a bacteria that grows big enough in size. It has enough cell material and everything else like that. What it will do, and enough DNA, and then what it will do, it will divide. And so it will basically grow, it will duplicate its genome, and then in the middle it will form a plate, and it will divide. And this is, so you have one bacteria divides up into two bacteria that have roughly equal amounts of cell wall, cell membrane, and all that kind of stuff. This is an example of Bacterium Salmonella typhimurium, which is one of the organisms that you should be looking at. In figure 9.3, we're kind of seeing a little bit how this happens. And so you have replication of the circular DNA. So once the bacteria gets big enough and everything else is all made, it will make its DNA. We then see a particular protein called FITZ protein that assembles, forming a ring around that's anchored in a plasma membrane. This then constricts and produces septum in the middle, where the peptidoglycan will actually be made, and then Z-ring pinches the cell envelope to separate the plasma into new cells with new peptidoglycan. And the cells are roughly equal to one another. So on this, we're going to get a doubling time. So typically you have a bacterium breaks in half, and so you'll go from one bacteria to two bacteria. And so in your first generation, you have one bacteria. After one generation, you now have two bacteria. After two generations, each of those two bacteria produces two bacteria, so you now have four bacteria. And so the equation for this, looking at this, is the number of individuals that you have in a population. It's on the lower right-hand side. And the number of individuals in a population is equal to, the number of equal population is equal to the number of individuals that you started with times 2 to the nth power. And so it goes 2, 4, 8, 16, 32, 64, and so on and so on. And that's called exponential growth. So adding cells to this. So what we do is the cells that you add. So when we add cells to a new growth. So let's look at some terms. So an inoculum is where you add cells to a new culture. That's called an inoculum. So please make me an inoculum. The food that you add is what we call the culture media. So the media is what the bacteria is going to be growing on and duplicating on. Generation time is basically what we call the growth rate. And so some bacteria can go really fast. E. coli can double roughly, given the right conditions, can double roughly every 14.5 minutes. There's certain bacteria that can actually even go faster than that. certain bacteria are really slow, like the bacterium that causes tuberculosis takes a very long time to grow, and it's very slow. And again, that's characteristics of the genetics of the microorganism that is there, and also the media that it's growing in. In this slide here, we're looking at how can we kind of model growth. And so this is what we call a growth curve. And so on this graph, on the left-hand side, you're going to have the number of cells, and on the bottom, you're going to have time. And so when you have cells are growing, your cells are going to have kind of a positive slope and be going up. When the cells are dying out, you're going to have a negative slope on there. And then what we can see here, because these cells are growing exponentially, we actually use a logarithm of living bacterial cells. And we'll talk about that in a second. But what I want to say here is that when we look at this, we actually have a number of different phases. And so number one, we have this lag phase. And so when you first put an inoculum into some media, the bacteria don't divide. Because what they need to do is they need to take the nutrients from their media, they need to make more cell wall, they need to make more plasma membrane, and kind of everything else. They got to get used to that media. So maybe they were growing on lactose before, and now you're growing them on glucose. Again, they got to get used to that. During log phase is when those bacteria are growing exponential. And so we know a lot about bacteria growing in log phase because that's one of the phases that we really like to have them go. And so what you see is those cells are dividing. They tend to be synchronous. They tend to all kind of divide at the same. And so in log phase, we have exponential increase in the number of living bacterial cells. In number three, this is a little bit harder area to study. This is what we call a stationary phase. And so what's always going to happen is that you have what's called a limiting factor on growth. And so when bacteria are growing up, they're either producing toxins, or let's say they use up all the glucose in their media, so they have nothing left to go. And so what's happening in that during that stationary phase, actually the cells don't really grow because their death rate is roughly equal to the division rate, or they're kind of not growing at all. And in level number 4, what we tend to see is that where the cells actually start to die off. Again, the cells are producing toxins and things like that, and so what we tend to see is where the number of deaths exceed the number of cells being created during that binary fission. And so what we can see is a lot of times, you guys won't be doing as much of this, but a lot of times I really kind of talk about students is that when we're doing biological tests on microorganisms, we really want to test things in the log phase. And that if a student left a bacterial culture in a test tube too long, that they would be in the death phase and the conditions would be slightly different. So these cells grow, these cells die. Sunrise, sunset. In this next one here, we're going to be looking at the plotting of cells. And so if we use an arithmetic scale, what's actually going is you have this thing that looks almost a lot like a J curve. And so what happens is that when we look at time in the arithmetic scale, you go from having 1 cell to 2 cells to 4 cells to 8 cells to 16 cells to 32 and so on, and it goes up exponentially. And what you can see between the hours of 1 and roughly 7, you can go from roughly 1 bacteria to 100 bacteria. Yet from 7 to basically 10 hours, you go from 100 bacteria to 1,000 bacteria. And so that's a lot, and so that will keep on going. How we look at these curves a little bit is we use something that's called a log phase, and so base 10, semi-logarithmic scale. And so in time, we're looking at usual time, and because we do the semi-logarithmic scale, actually we're looking at base 10. And so those first couple of hours, 1 to 7, we go up to 100, and then this flattens out this curve a little bit, and it's much easier to look at. Okay, and we'll explain that a little bit more. How do we maintain a growth cultures? And this cartoon that we have in here is something that we call a fermenter. It's actually this particular one here is a chemostat or a bioreactor. And Genentech, which is just down the street from us, has a number of these. But if we look in the past, and we're looking at this growth curve, so maybe we want a bacteria at this point in the growth curve. How we can get that is by growing things up in the chemostat. The chemostat is a culture vessel fitted with an opening to add nutrients feed and an outlet to remove contents effluent. And so what we see is we can design it so that we're adding bacteria, so we're adding our media in here as we're moving it out, and so we can control what phase of growth this is in. This is also what we call continuous culture as well, and we can control the amount of oxygen and everything else in here, and we mix up this vessel in here. And so this is a great way to grow up microorganisms and control at what stage in their growth they are in. So that being said, we need to know how many bacteria are in a culture. How do we do that? And one of the ways that we have to do that, and again, this is a way of looking at things like blood, etc., we can use a counting chamber, a Petrov-Hauser chamber. And so in this chamber, we have little squares that we look at underneath the microscope, and we know the volume of that square. And so if each square is, let's say, 1/125th of a mil, we know how big it is, we count 10 bacteria, and we know that there are that many per mil in here. And so I'm not going to go over the math right now. We'll do that a little bit later. But in what we call a direct measurement is that we can put the bacteria in a slide and we can count them, even if they're very high in number. Another way that we can count living things is we can use something that we call fluorescent staining. And so we can use the fluorescent staining to tell the difference between living cells and dead cells. That's a problem. When you're looking at it under a microscope, cells don't do a lot. They're not going to wave back at you and say, hey, I'm alive. And so you can add certain things like trypan blue or fluorescent staining. And what happens, these kind of dyes are taken up by dead cells, whereas they're not taken up as much by living cells. And so that way you can tell the difference between things that are alive and things that are dead. Now this counting of cells is pretty laborious and it's kind of hard to do, and some of you may do it, but can we automate that? And so there are things that we call a Coulter counter, and in a Coulter counter we have a sensor, and that what we can do is add, we have a small little hole, and that as bacteria move through this hole or a particle moves through the hole, it actually changes the amount of electricity that goes through there, and we could count that. So we could run a sample through there and we could count that. And we've been doing that, for a long time. Another way of counting cells, and again, this is another way of counting viable cells, is what we call a serial dilution. And what you do when you do a dilution, you kind of do it all the time, right? And so when you're making orange juice at home, you have that little container of orange juice, and you're supposed to add 2 other containers of water to that container of an orange juice, and basically you get a 1-to-3 dilution. Now imagine, if you will, is if you have 1 billion cells, 1 times 10 to the 9th cells, and you need to dilute it out, you would have to take a little thimbleful and put it in a bath of a swimming pool in order to get that dilution that you want. However, we can use something that's called a serial dilution. And so in this case here, we take our original culture of bacteria, we dilute it out 1 to 10, and when we count that out, we still have too many bacteria. But again, if we take that 1 to 10 dilution and now we dilute it 1 to 10 again, that's a total 1 to 100 dilution. We're starting to see individual colonies. We do it again, we get a 1 to 1,000 dilution. We do it again, we get a 1 to 10,000 dilution. We do it again, we get a 1 to 100,000 dilution. And so at a 1 to 100,000 dilution, we're actually able to see a million cells, right? So if we pull this out, we would have a 1 to 100,000 dilution. We would have 10 cells if we had 1 million cells per mil. Again, we're going to talk about this a little bit, and we'll have exercises on this as well. Bacteria are a little bit different. We have a number of ways of doing that dilution. So in the previous set here, that's what we call a spread plate. We would spread the bacteria out on it. Bacteria don't necessarily grow there all the time. Another test that we have is actually by doing the dilutions in melted agar, and then we would swirl up the agar and we'd pour out the agar, and we could take a look at, so bacteria that like to grow again within something would be able to grow. And this here is looking at the spread plate method. Again, we would typically do that in class. Another way that we have of looking at things is looking at statistical measurement. And so you have the most probable number method. And so in this case here, you can have a relatively small number of cells, and you can have an idea of how many bacteria that are on there. And so what I want you to know is just in the back of your mind, say most probable number, and we'll talk about that and have an exercise on that a little bit later. Indirect cell counts are, what we just talked about now were basically direct cell counts. You're actually counting one bacteria. Count of one is one bacteria. In indirect counts, what you can do is you can measure other parameters of your culture to get an idea of how many are in there. And so you get, again, you do indirect cell counts when you cannot count the cells directly. And so if you're looking at bacteria that you can't necessarily grow or it will take too long, etc., there's a number of different things. And so we can look at turbidity or cloudiness. So you can use a spectrophotometer. We can shine light through it to tell how many bacteria are there. We can dry down our bacteria and see how much that we have. In metabolics, we can actually look at the results of metabolic processes. And so again, living things are using glucose and living things are producing carbon dioxide. And so we can measure the amount of glucose that's used by a culture, and we can measure the amount of carbon dioxide that's produced by a culture, and that gives us an idea of how many that are out. This is looking at a spectrophotometer. You guys may have seen this in lecture or other biology classes. And so here we have a culture that doesn't have a lot of bacteria in it. When we shine a specific wavelength of light through here, there's no bacteria to absorb this particular material, and so we're going to get a lot of light out. In this one here, we shine a particular wavelength of light through there, and again, sometimes it's about 420 nanometers. That light is scattered or that light is absorbed by the microorganisms, and we get much less here. And so that ratio can give us an idea of how many bacteria there are per mil. We would set up a standard curve, and that we can do this. Again, not all bacteria divide by what we call binary fission. Okay, sometimes we have fragmentation, so especially things like fungi, or especially things like filamentous bacteria or algae. What you can have is where you break a piece off, and that piece can become a whole nother organism. In budding, what we see here, yeast tends to do a lot of budding, is you may not want to give, when you divide equally, each bacterium has an equal chance of surviving, right? Because they have the same amount of material. But what you may see is that certain organisms, what they do is their offspring, you have a genome, you have everything that you need to grow, but I'm not going to give you all of my material. I'm going to keep that. So you produce a bud, a smaller form, a mini-me of yourself that goes out and that hopefully is able to grow. In number 9.17, we're actually looking at what we call a biofilm. And in a biofilm, you have a mixture of different bacteria that are growing together in this biofilm. If any of you have seen kombucha, that mother that they have is actually a biofilm on that. And so basically you have kind of reversible attachment. The bacteria can attach to something. They produce this EPS, this extracellular polymeric substance that kind of allows them to bind to that. And then the bacteria can kind of move or swarm or go on, or they can last for years. The thing with these EPSs and these biofilms is they're resistant to drying out. They're resistant to antimicrobials, antibiotics, or certain other drugs that will prevent them from working. Again, in sewage treatment plants and stuff like that, these actually form films on the outer side of the pipes where organisms live there, and so ideally they take nutrients that are coming off. Why biofilm? It's a symbiosis, so we see that the microorganisms are stronger, kind of working in unison with one another than they would be individually. You can reduce predation, and so a lot about predation is how big you are. And again, organisms that are smaller get preyed upon a lot easier. But those organisms bind together, then they're less likely to going to get preyed upon. This also provides, again, protection from the environment, and I talked about drying out, UV radiation, all those kind of things. And again, what we call the dispersal form of this is what we call a platonic form. And so the bacteria that can float around, those bacteria will come together and form the biofilm. Cell-to-cell signaling is important in what we call quorum sensing. And so, again, what's going on with quorum sensing, and so I'm showing you here gram-negative. So basically, each cell produces a certain chemical. And so it's a short, in the gram-positive cells, it's this kind of short, weird-looking peptide. In the gram-negatives, it's N-acylated hormone, homoserine lactone. And so what we see is the bacteria produce this at a certain rate. And what's going to happen is the more bacteria that you have, the more of this is going to be produced, the higher concentration. So that way, in quorum sensing, basically what a quorum is a certain limited number of people that you need in order to vote on something or that. And so the same thing with bacteria. It's like you need, again, if you send out 1 or 2 bacteria, they're just going to die. Yet if you send out 1 million bacteria, that swarm would be better at survival. And so, again, the bacteria can't see. They can't see how many are there. They can't hear one another. But what they can do is they can produce a signal molecule that they can then respond to. So that's growth. We're going to look at next at some requirements that cells have of growing. And so remember, when we looked at that growth curve, is that in order to have exponential growth, you have to have enough oxygen, carbon, nitrogen, temperature, kind of everything else like that in order for those conditions to go. And so what I want you to learn, so what are your learning objectives for oxygen? So you need to interpret data for oxygen requirements, know the categories of oxygen requirements, give examples of microorganism and their importance to human beings, some of these oxygen ones. And so, again, just to give you a little history, oxygen is a very reactive molecule and actually can kill bacteria. And bacteria actually need to have a way to survive the presence of oxygen. And so just like oxygen will oxidize your car and rust your car, it will oxidize the molecules of your cells. And so if a bacterium cannot respond to oxygen, then it's not going to survive. And so basically what we think is a long time ago that the Earth had actually very, very little oxygen in the atmosphere. There's very little oxygen there. There's oxygen in rocks and in water and everything else. But in the atmosphere, there's very little oxygen. And so what we see, looking back at it, there are certain conditions on Earth, the bottom of ponds where there's a lot of rotting, or in the gut of ruminants, where we have this what we call anaerobic, without oxygen atmosphere. And there's lots of bacteria that actually grow in that atmosphere. How we can do that is usually by kind of altering our growth conditions. And so we talked about there not being a lot of bacteria not being able to grow under a number of conditions, but in this one here it is. So can we see that? Yes, we can. We actually use thioglycolate. So thioglycolate on the left-hand side is the non-oxidized form. What can happen in the presence of oxygen, you can actually get these, so the hydrogen gets scraped off and you form this disulfide bond here, and that kind of scavenges oxygen. So if we make actually media with thioglycolate inside of it, it will actually kind of remove the oxygen from tubes. And so if you have what's called obligate, you have an obligation. If you have an obligate aerobe, it will only grow in the presence of oxygen. And so in this media, down a little bit deeper in the media, all the oxygen has been taken up by the thioglycolate. And so what we see is microorganisms will only grow at the top. If we have what we call an obligate anaerobe, these organisms will only grow on the bottom because, again, there's a little bit of oxygen on the top. If we have what we call facultative anaerobes are able to grow in the presence of oxygen, but they're able to grow in the absence of oxygen. So they have the biochemistry that would allow them to do it. Again, the growth is going to be much better in the presence of oxygen. In the absence of oxygen, you will still have growth, though, in the absence of oxygen. Your muscles kind of work the same way, and I'm not going to get into that. Some of the obligate aerobes include the Mycobacterium tuberculosis that causes tuberculosis. Obligate anaerobes would include organisms like Bacteroides that we find in the gut and in fecal matter. I'm not going to tell you the rest of it here, but facultative anaerobes have this particular growth pattern. Aerotolerant anaerobes, and what we see is aerotolerant is that it will grow in the presence of oxygen. It has a way of mitigating the damage that's done by oxygen, but the great thing when we see this is we look at it in the presence of oxygen. In the absence of oxygen, it grows the same. Okay, microaerophiles are actually organisms that they tend to grow in low oxygen environments, and so in these thioglycolate tubes, we tend to see it a little bit off of the surface of the tube. And so what you may want to do is, in your reading and your encounters with microorganisms, see which fits where. So with all this oxygen around that we have, and the thioglycolate is good, but sometimes we want to look at microorganisms. How do we grow those up? And so we can use this a thing called an anaerobic jar. We can grow up the bacteria, and what we do is we put some little packs in there, and we can remove the oxygen. We can use an anaerobic box, and so we would flood this chamber with an atmosphere that we know, an atmosphere without oxygen, and then we have gloves, and we can do our manipulation of materials inside of there. Some notable anaerobic organisms that we see would be Clostridium perfringens, that causes gas gangrene. And so here's an individual, probably had diabetes or something like that, had reduced circulation into the feet, and we tend to see these get really gnarly. That was probably cut off. Ugh, don't want to even think about that. How do a real simple test that we would have for detoxification of oxygen is what we call the catalase test. And so you can actually, and this is a biochemical test, and this is one of the things that we know. So when we do a dichotomous key, we would do that. And what we see in a catalase test is you made peroxide is actually used to kill actually a lot of bacteria. So the peroxide, in the presence of this enzyme called catalase, will actually be reduced to just water and oxygen. That's what these bubbles are. And so the catalase gets rid of that. So the catalase can be detected in the presence of those particular enzymes. And hopefully we're going to have a lab on that. All right, so the oxygen. The next I want you to look at is the effects of acid, pH. Again, you need to be able to describe the various effects of pH on a culture. You need to define the categories, and you need to give me examples of economically important species. Now, pH is very important because a lot of things that we like actually have a pH that's close or a little bit below 4. And so things like yogurt, pickles, sauerkraut, and that's ceviche are foods that actually use acids to promote the growth of good bacteria and to inhibit the growth of bacteria that we do not want. And what this is showing you here is that this is the pH curve, and so we have going from 0 to 12. Again, we're looking at I think it's the inverse pH. So in 12, there's very little hydrogen ions present. At 1, there's lots of hydrogen ions. An acidophile is an organism with a pattern that it tends to like to grow in acidic environments. A neutrophile tends to grow in neutral environments, and an alkanophile tends to grow in alkaline environments, typically above 9. Those are the characteristics. Again, if we view these cultures can be very huge. In this one here, there's a lake in Tanzania that is alkalophilic, so it's fed by streams from volcanoes, etc., and lots of salt and lots of base inside of there. And so probably you'd put something in here and it would eat you up like a battery acid. But again, there's microorganisms that grow there, and these microorganisms have these kind of green colors, and you can see those through a satellite. Again, at the bottom of the ocean, we have these things that are called smokers, where we actually have water coming out through volcanic activity. And if you look on here, there's lots of bacteria and stuff that can grow in that under those conditions. And so these are little worms, and the feather worms, and they have a symbiotic microorganism with them, but they also eat the bacteria. And so as these chemicals are leaving here, you have microorganisms that can use the chemicals of those organisms to survive. And so what was really kind of interesting, for a long time we thought all of life on Earth depended on sunlight. And this shows you that you don't necessarily have to have sunlight in order to have these reactions occur. And we'll talk about those a little bit later. So for temperature has a big effect on microorganisms as well, and we'll look at the requirements and the effect of different categories. And so again, when we look at temperatures, we tend to look at these things that we call mesophiles. A lot of our pathogens grow in us. They tend to like the same temperature that we like, maybe a little bit higher, maybe a little bit lower. The psychrophiles are ones that like it really cold, like in a refrigerator or on a tundra. Thermophiles and hypothermophiles tend to like it a little bit warmer. And so the microorganisms that can live next to those thermal vents are these thermophiles. And so again, have a general idea of what's going on with these guys. Other physical and chemical effects, nutrients that are in a culture will affect whether a bacteria is able to grow or not. We can also look at osmotic and barometric pressure. We can also look at the presence of light. So these are all things that can have an effect on growth. In class, we're going to talk a little bit about media, and so we can talk about enriched media, which has a lot of really good stuff in it. We can talk about fastidious media, or organisms that are fastidious that actually don't require a lot in order to grow on. I will talk about chemically defined media. That's where we know exactly what chemicals are in the media. We can talk about complex media. So again, we grow things on nutrient agar. So nutrient broth is basically beef broth. And then we can have an idea of what's in beef broth, but we don't always know exactly what's there. In selective media, we're looking at media that will only grow, certain organisms will grow. And so if I want to grow bacteria that were originally growing in pickles, I may want to try a media that has a low pH, right? Because all the other microorganisms won't grow, and that would select just for the growth of my organism. Enrichment is basically where you have something. So if I wanted to isolate some bacteria that would eat fuel oil, what I may do is put a low concentration of fuel oil into some media, and so that way my bacteria would actually grow, and I would enrich for that. What we call differential media is where one bacterium is able to grow on another. And so again, if you had a bacteria that was not able to use glucose, if you put glucose in the media as the carbon source, it wouldn't be able to grow. That's it right now for, again, chapter number 9. You've had a good introduction onto how cells grow, a good introduction to know, again, this cell growth curve, and then we talked a little bit about the conditions that promote growth of microorganisms. This is Nick Cap saying over and out. See ya later.